You are welcome to Senior Teleclass on Tek TV. My name is Kwaku Frimpong. This is our first lesson on biology, SHS 3. Our lesson is on nucleic acids. By the end of the lesson, you should be able to understand the term nucleic acid, types of nucleic acids, the structure of a nucleotide, differences between ribose sugar and deoxyribose sugar, types of bonds in nucleic acid. Now, when we say nucleic acids, it's a chemical which is found in the cell of every living thing. And this chemical is unique to a particular living thing. Basically, it's made up of three main components, which are a sugar, which is called pentose sugar, a nitrogen-containing base, and a phosphate ion or phosphate. These three components together will make up a nucleic acid. However, this nucleic acid is a chain consisting of the pentose sugar that we mentioned, the nitrogen-containing base, and the phosphate. And each particular chain is called nucleotide. So the chains that make up the nucleic acid or nucleic acids are called nucleotides, consisting of the sugar, the nitrogen base, and the pentose, and the phosphate ion. So we can say the functional unit of any nucleic acid is a nucleotide, or the functional units or the repeating units of a nucleic acid is a nucleotide. So if you should divide a nucleic acid, we're going to get portions that we will call nucleotides, which are repetitive. So a nucleic acid is a chain of nucleotides. Once it's a chain, then there should be a bond between the chains or between one chain and the other. And these bonds will be formed by a portion of the nucleotide where we have the phosphate, and then a portion on the pentose sugar where we have the hydroxyl ion or the OH by a dehydration reaction, which means that water will be formed as a result of the bonding between one nucleotide and the other. So the types of bonds are two. And they are the phosphodiester bond, which is a covalent bond formed by the phosphorus or the phosphate ion, and then two ester bonds. So the term diester means that there are two ester bonds with the phosphate or the phosphorus in between. By so doing, one nucleotide will be able to join to another nucleotide vertically or horizontally based on how you are looking at it. Then, like a ladder, comparing the nucleic acid to a ladder, there is another bond which is called the hydrogen bond, which we shall see where it is found as we progress. So now, by looking at the picture, we can see where the phosphodiester bond is. So from the picture that we have here, you can see the phosphodiester bond over there. So from my picture, I can say this is a vertical bond Then I can see one of the hydrogen bonds also there, which is a horizontal bond from my picture. So based on the orientation, you can determine to say that the phosphodiester bond is a vertical or horizontal bond. But from my orientation here, I can see that the phosphodiester is a vertical bond and the hydrogen bond is a horizontal bond. Now, having known that a nucleotide consists of pentose sugar, phosphate ion, and a nitrogen base, we want to look at their structure so that we can easily identify them when we come across them. So that is the nature of the phosphate ion, the nature of the sugar. There are two sugars which we should take note of by looking at where the OH bond is found on my left. Then we look at the other bond on the right, down there. So we can see that on my right is where we have the ribose sugar. That one has lost one oxygen. That one has got all the OH over there. Then on the left, from the name deoxyribose, we can see that there's a loss of one oxygen over there. That is where the difference is. So whenever you have to write the structure of a deoxyribose sugar and a ribose sugar, you should note that the difference will be centered on where we have the H on my right and where we have the OH on my right. Then the bases are also in two forms, those with double ring and those with single ring. That is what we need to know, so that whenever we see those with double ring, we know that they are the purine bases. And those with double ring will be called the pyrimidine. So the purine bases, we have the adenine, denoted by capital A, the guanine, also by capital G. Then the pyrimidines, the single rings, thymine, uracil, and cytosine. They are the single ring. Take note. So the difference is the ring. Here, only one ring, and the purines, double rings. So the one rings we can see here. Then we can see the double rings also here under the purines. So when we put all the three components together, diagrammatically, we can get this as the functional unit of the nucleic acid, which is the nucleotide. So anytime you have to mention the components of a nucleotide, then you mention the phosphate, the pentose sugar, then the nitrogen base, which can be in different forms, as we've mentioned. So detailed structure of what we just showed is also here. Deoxyribose with H on my right on the second bond. Then this one has got OH also on the second bond. So that's where the difference comes from. They are all having bases and then phosphate. Now, how do we relate chromosome to nucleic acid and then gene? It's very important that we know the differences. So in a cell, we can have the center called the nucleus. Inside this center, we have the chromosome, which is the carrier of genetic information from one generation to another. Good. So within this chromosome are what we call the genes. Then what is a gene? Then we come to the nucleic acid. So if I have this as my nucleic acid, for instance, then any portion of this nucleic acid qualifies to be called a gene. So it means that the nucleic acid that we are studying today are made up of specific portions which we can call a gene or genes. Portions of the DNA or the nucleic acid are called genes, and these portions are found within the chromosome. So basically the chromosomes are just carriers of our nucleic acids. Then when we cut the nucleic acid into pieces, then we get our genes. So that's the relationship between chromosome, nucleic acid, and a gene. Now there are two examples of nucleic acids. The DNA, which is the most common one. Of course, we can see that the DNA, which is an example of nucleic acid, is having a deoxyribose sugar. Hence the name deoxyribonucleic acid. It is one of the examples or the types of nucleic acid. Then the other example or type is the RNA, made up of a ribose sugar. So you see the type of sugar has a lot to do in terms of identifying the type of nucleic acid. So the name ribose sugar is reflecting in the name ribonucleic acid, which is the RNA. So the two types of nucleic acids are DNA and RNA. What are the differences? The differences between DNA and RNA. There are so many differences. We've already mentioned some of them. The type of what? Sugar. Yes, under DNA, the sugar is deoxyribose. Under RNA, the sugar is ribose. Remember, we said the difference is based on the second carbon, the structure on the second carbon. That is where the difference is coming from. Then the type of base, which is basically what makes one block or chain of the nucleic acid different from the other block or chain. As we said, the nucleic acid is a chain. So one chain will be different from the other chain, or one block will be different from the other block, or one region or one portion of the nucleic acid is going to be different from the other portion based on the type of base. Because the DNA will have a phosphate, it will have a pentose sugar. The RNA will also have a phosphate, the same pentose sugar, except that the second carbon bond will be joined to a different structure in the RNA. But the main difference between one nucleotide or one functional unit of a nucleic acid and the other is centered on the type of nitrogen base. So the second difference is that a DNA consists of a pyrimidine base, which is cytosine and thymine. And then RNA consists of a pyrimidine base, which also is made up of cytosine and uracil. So it means that the two bases here, we can see cytosine also found in DNA. So you don't have any problem with that. Cytosine in RNA, cytosine in DNA. But uracil, common to RNA only. Then thymine, common to DNA only. Good. So the difference is uracil here, thymine there. as pyrimidines. Then the function of RNA is normally in inheritance, hereditary. Then that of function of DNA is in hereditary. Then the function of RNA is on protein synthesis. So inheritance, characters are carried from one generation to the other under the instruction of the DNA in the organism. Then proteins are synthesized by using the RNA. A DNA is found to be stable because it's always there in the organism. And then the RNA happens to be highly unstable. It keeps on changing based on the function that it is performing at a particular time. DNA happens to be a double structure. So it's something like this. Then the RNA happens to be a single structure, making the DNA heavier than the RNA. So the molecular weight of the DNA is relatively larger than that of the RNA. These are some of the common differences that we can write between DNA and RNA, the two main examples of nucleic acid. So as we mentioned earlier, the differences is much centered on the sugar and the bases. So if you cannot remember any of the differences at all, these two are very common to come by. Having mentioned the bases under the differences, let's see the types of bases. Under two categories, purine bases made up of adenine and guanine. Then the pyrimidine bases made up of cytosine, thymine, and uracil. So you see why we are putting the first letters in brackets, because the bases are where the differences between one nucleotide or one chain of a nucleic acid come from. So very soon, a DNA structure or an RNA structure can be represented by just the bases, because we believe that all of them are having sugars and they are all having phosphates. So the main differences is centered on the bases. So we will be writing the DNA structure later using the alphabet in the brackets. That is why we've structured them like that. So we should always remember that the alphabet in front will go with the names of the type of bases. So the bases are used to represent the entire DNA or RNA structure. Is it true? Then we want to look at how the bases are paired. Because the pairing of the bases will show the structure of the DNA that we are looking at, or the nucleic acid that we are looking at, or the RNA. They do not pair anyhow. So Chagaff, a scientist who came by the arrangement, came out with a rule which is saying that the group that we call the purines, made up of adenine and thymine, will always have a proportion which is equivalent to the purines, adenine and guanine, will have a proportion which is equivalent to the pyrimidines. Remember we said here that the purines are one group. The purines are one group. Then the pyrimidines are also another group. So we have adenine as a purine, guanine as a purine, cytosine, uracil, thymine as pyrimidines. So what we are saying here is that the purines will pair with pyrimidines, basically. So adenine, which is a purine, is going to pair with thymine, which is a pyrimidine. Then cytosine will also pair with guanine or guanine. No two purines can pair from the rule or from the law or from the base pairing. No two purines can pair. No two pyrimidines can also pair. So basically, purines will pair with pyrimidines. Then specifically, adenine will pair with thymine. Cytosine will pair with guanine. So, based on the rule, always we will see that the bond between A formed by A will be connected to T, that is thymine. Then the bond formed by G, guanine, will be connected to C, cytosine. The bond formed by A will be connected to T. Then the bond formed by G will be connected to C. So you see on the screen, thymine there bonded to adenine, then guanine there bonded to cytosine. So G to C, then T to A. Remembering that the bond also is triple here, that's between G and C. A triple bond here, then a double bond there between thymine and adenine. Remember, the orientation can change. It can be adenine and thymine, it doesn't matter. It can also be cytosine and guanine, it doesn't matter. But remember, the bond is triple between cytosine and guanine or guanine, then double between adenine and thymine. So we look at what a nucleic acid is. Then we also look at the types of nucleic acid, which are DNA and RNA. Then we look at the differences between them. And we said one of them has got a ribose sugar, the RNA. Then the DNA has a deoxyribose sugar. We mentioned the types of bases, purine bases, pyrimidine bases. The bond between the bases. We said the purine will always bond with the pyrimidine. And no two purines can pair, no two pyrimidines can also pair. And we said that based on that, the bases can be used to represent DNA or RNA. So here ends our lesson for today. Hope to see you another time.